Hello, I'm Dave Tabb, and today I'd like to take you through some of our fun work, very cool work, combining transcriptomics and proteomics in the world of chia, a plant that we hopefully all will be eating a little more of soon. Here it is growing in the field. This is work that I've carried out in collaboration with collaborators at the University of the Western Cape, at the USDA, and even at Massachusetts General Hospital. So it was an interesting team of people coming together on one project in plant proteogenomics. I'm going to talk today about the combination of data from 4 different studies carried out by RNA Seq in Chia, the Salvia hispanica, and 3 proteomes for that same species. So trying to find the best way to combine all of these data to tell one composite picture. This work was very recently published in the journal MD-PI Plants. In the description to this video below in YouTube, you should be able to see a link to the paper as published and also to these slides available in PDF format. So we'll talk about the combination of these different data types as well as some takeaways for plant biotechnology that came out of this work. So first off, we might ask, why is chia a superfood? What is the thing that makes chia so interesting? And I think that there are lots of reasons for why we would want to study chia from a nutritional point of view, but probably number one is its ability to create polyunsaturated fatty acids. I'll probably call them PUFAs along the way. But our body doesn't make much in the way of polyunsaturated fatty acids, and we need to eat them. At the moment, the way that most people get polyunsaturated fatty acids is through eating fish. But even if we eat all the fish in the ocean, we are not able to feed the whole human population of Earth in a sustainable way. So if we can grow species that generate polyunsaturated fatty acids instead, it's a much better option for the future of humanity. Now I'm going to be talking about RNA-seq quite a lot. I know some people who follow my channel are mostly interested in proteomics, so it might be that RNA-seq is a little unusual for you to be thinking about. So I would just note that there are 3 major ways that we use RNA-seq routinely in biotechnology. The case shown here on the right is the idea that we have a known genome sequence, we've annotated where the protein coding genes are, and we know where the intron and exon boundaries are, and so on, and then we use RNA-seq to say which of these possible transcriptions for a gene are being produced, and do things like differential gene expression and so on. We're at the other extreme here, though. We're all the way over to the left. We have no genome sequence for chia. We have no gene models for chia. Instead, we have just RNA-seq data, and we need to be able to patch those together to assemble these tiny little bits of sequence, these reads, into contigs. And here those contigs represent transcripts. So from sequencing data, we're trying to produce intact transcript sequences. Now, how did I know that chia was going to be a real problem for us in proteomics? Back in 2017, my friend Lysax came to me saying, look, I've got these data I've produced, these LCM-SMS data from chia, but how do I identify them? When we looked up Salvia hispanica in NCBI's taxonomy browser, we saw that the number of sequences listed for the species was negligible, 302 protein sequences in total. That's obviously nowhere near what the chia has on offer. But we saw that there was a genome listed for NCBI, and we were really excited by that until we clicked through and realized that the only genome available for the species was from its chloroplast only. So the nuclear genome was just not measured in this area. On the other hand, we saw that there were SRA experiments. These are the sequence read archive, sometimes called the short read archive. What we see is that there were 68 different experiments from sequencers that had been reported for this, and that they grouped under 8 different projects. So we were able to get quite a bit of information about Salvia hispanica, but it was going to be in pretty raw form, not finished in terms of, here's a list of sequences for you. Now, these are the 4 projects that I'm really focusing my attention on within chia. Sridhar was the very first one published. They used a Genome Analyzer 2X. And if you're not familiar with sequencer technology, I would just say that this is the closest relative to an instrument released by Solexa back in 2006. Now, people who know anything about sequencing will say, Sequencing technology changes all the time, so why would you even bother with something that came off a mass analyzer from the middle of the first decade in this century? That's ancient times from the sequencing world's perspective. But it was what was available at the time we produced these data in 2017, the proteomics data. Pelaez had a very different kind of study from Sridhar. So Sridhar focused on just developing seeds. Pelaez was looking at sequence variability within the species. So they had 4 different cultivars of chia and several different wild types that were growing in the wild, I should say. So they had seeds measured only, whereas Sridhar had used the early development of seeds, which will reflect different genes being expressed at those 2 times. Wimberly set out to create the first reference transcriptome for this species. They used 3 shots or 3 lanes of leaves, and they used 3 lanes of roots for these plants, and then combined all of that data together in one transcriptome. They used the newest type of mass analyzer within this study, the HiSeq 4000, which is the same as what Pelaez used, and they generated these data and assembled them and released the assembled sequences that they produced from this experiment. So that was lovely. We could ask whether our techniques for assembling sequences were comparable to the ones that were used by the authors of the data set themselves. Now the data set that shows great promise is this one from Gupta. A team over at Oregon State did this lovely collection of 13 different tissues of chia. And our feeling was, gosh, if a gene gets expressed anywhere, we've got a much better chance of seeing it if they've analyzed lots of different tissues. But they did not have access to the HiSeq 4000. They were using a slightly older model called the HiSeq 2500. So you can see that the HiSeq 2500 is able to produce only about 1/8 as many sequencing reads as the instruments used in these middle 2. So which of these experiments is going to be the best bet for us? We weren't sure. We had our assumptions that if you produce more reads, you have a better transcriptome. We had the sense that if you produce longer reads, say 100 versus 72 base pairs, you might have a better transcriptome. We thought if you have more tissues represented in your sample, that might give you a better transcriptome. But these things all sort of balance against each other. How will we determine which of these is best? Now, if you're familiar with RNA-seq but not with proteomics, I would just point out that we start with complex protein mixtures, maybe 10,000 proteins or more in an individual sample. We digest those to peptides because peptides are a lot easier to deal with in the biochemistry and analytical chemistry downstream. We often fractionate by separating this wildly complex mixture of 100,000 peptides to different aliquots. Based on some biochemical properties, we make it easier to get a comprehensive, as comprehensive as possible list of peptides from those samples. We use LC-MSMS, which is described by these other icons, meaning that we use reverse phase liquid chromatography to separate peptides. We use electrospray ionization to make ions of those peptides, and then we measure both the intact masses of peptide ions and the tandem mass spectra, which is to say the fragments produced from blowing apart each peptide. So this is the basic data frame for how the proteomics data get generated. So we had 3 different proteomes to work with. The original study, Husselman 2017, produced a whole bunch of raw files for leaves that were from plants under different stress conditions, caffeic acid or salt or other things of that sort. Cooper was a data set produced explicitly for this manuscript. So my friend Brett Cooper produced a proteome of roots, of hypocotyls, of seeds, and cotyledons. But due to a miscommunication, he made use of S. columbariae, not S. hispanica. The reason for this confusion is one that shows up a lot in plants, which is to say that both of these species get sold as chia. And because of that miscommunication, the sequences he was working from were going to be somewhat different from Salvia hispanica. And the third data set was one that had been published in the last year or so by Aguiar Tuala. And in their work, they produced just a single LC-MS experiment looking at the extent to which seed proteins of chia are able to resist the actions of proteases, in this case flavorzyme, and alkylase. So it's a very difficult raw file for us to work with because it's got because any peptide could start anywhere in a sequence and end anywhere in a sequence. But helpfully, they made their data public for us to use in this paper, so I really appreciate that. Open science is a big theme here for all of these works. How would we be able to have undertaken these studies without the data becoming public? So the three questions that kind of motivate everything in this project would be, where can we get protein sequences for LCMS and for LCMS-MS identification if the repositories like NCBI, like UniProt, have essentially nothing for our species? If the database doesn't have it, how do you work with the data at all? We're so accustomed to having those full-length protein sequences available. Are transcripts assembled from sequencing reads sufficiently accurate to match observed peptides? And to just drive this point home, if the RNA-seq experiment tells us that the amino acid at this position should be valine, but in our cultivar we had leucine, for example, that would prevent the correct peptide from being associated with this tandem mass spectrum. So we need the sequences that we assembled out of these reads to be useful. Finally, if we could translate protein sequences from the ones that we've assembled in the transcriptome, could we determine functions for any of these? It's lovely to be able to say, this spectrum represents this peptide from this protein accession. But if at the end of the day you only know it's a protein accession and don't know anything about what the functional parts of that protein are, or even better, that it has an ortholog that's well characterized in another species, it would be pretty frustrating. We want to be able to know that having identified a protein, we have some idea as to its function. So these are three very different questions that are all quite difficult to work with in this class of data. So I'm going to try to tell that story through a series of different vignettes. We're going to start with how do we assemble reads that we download from Sequence Read Archive into transcript sequences. And for us, that answer is Trinity. How did we align reads that we had from these experiments to these assemblies to tell us something about the gene level support for on the basis of how many reads do we have for these different transcripts? For us, salmon gave us the answer. Can we compute a completeness metric for our transcriptomes? For that, we're using BUSCO, which focuses on a set of genes that should appear in just once in genomes of these species. Can we seek orthologs in the transcriptomes or proteomes of nearby taxa? For us, we chose the software Protein Ortho for this purpose. Can we check for known sequence motifs to help us annotate what these proteins actually mean? For us, InterProScan was what we used. And finally, can we match MS-MS scans to translated transcript sequences? And for us, MS-Fragger gave the answer. We also used MSGF. Okay, so first off, how do we build transcript sequences from reads? This FASTQ format is what you end up downloading from NCBI. Having done that, one of the first steps people frequently use is called FastQC for doing a quick quality control of the FASTQ files that have been produced. So that's a very handy piece of software. Frequently, people will also use trimming, and they're trying to accomplish one of two or both of two things for that purpose. First, they want to remove sequencing reads that are so low in quality that they couldn't possibly contribute anything of value, or to be able to trim off the ends of reads if those end pieces are of too low a quality. And the other thing they want to do is remove adapter sequences. Adapters are bits of DNA that allow you to, for example, PCR up many copies of the inserts that you're going to sequence. Okay, so having done all that work, we can then use Trinity to turn this collection of FASTQ files that have been cleaned up into an assembled transcript sequence set. Now, I've written FNA here. I don't want to confuse anyone. That is a FASTA file in nucleotide format, not amino acids. Now, this Trinity step is really a difficult one for a lot of labs to get moving on because installing Trinity has a lot of dependencies, and you must do it under Linux, and you must do it on a computer with a lot of RAM. And you might be thinking, well, I've got a computer with 16 gigs of RAM. That's good enough, right? And the answer is not even close. To do a proper trinity assembly, you're really going to want typically 64 gigs of RAM or higher. So you might need specialized computers to do this. If you're doing this professionally, you're probably going to be using a computer with 128 gigs or 256 gigs of RAM. So that is a limiting step for a lot of people. When we did this with the different RNA-seq data sets, we found a huge disparity in how many different transcripts were produced. In Gupta, we had a huge number of sequences that assembled. Wimberley, a huge collection. So both of these guys were setting out to do a transcriptome for the first time for these species. So lots of good sequences here. Sridhar, the earliest of the data sets, looks like a pretty bad transcriptome just from the total number of transcripts. Now I'm going to come back to that and explain to you that in fact the Sridhar transcriptome is very good. But I want you to note at this point that the assembly of those sequencing reads gave us very small numbers of transcripts compared to everything else. Now Pelea's has 4 different bars here, and the reason here is that there were 4 different cultivars that they considered in 2 or 3 sequencing experiments apiece. And what we see is that the number of transcripts that we could assemble from Pelea's Celea, a particular Chia cultivar, is more comparable to what we saw from the other 3 experiments than Pelea's Kualak, Pelea's Jalisco, and Pelea's Veracruz. So we chose the Pelea's Celea for consideration. We generally ignored the other 3 cultivars because this first one was so much better in terms of number of transcripts assembled. We wanted to see how many reads supported each of the transcripts that we were able to build from this. So for this, we use the Salmon algorithm. It's part of Trinity already, and Trinity has a helpful script to allow you to align a FASTQ file against the transcriptome that you've just assembled, so we just use that. So for each FASTQ, we did an alignment of those reads via Salmon to the transcriptome that we had assembled for that species. This gave us measurements of how many reads were seen per transcript for each FASTQ. We could then merge those results together into one report to say, in each of these FASTQs, how many hits did we have on each transcript? Now, I want to highlight something from Gupta here that I think will be a little surprising to some folks. We often think, okay, well, in proteomics, we have this big problem that the mass spec generates lots and lots of peptides from a few concentrated proteins, but produces fewer spectra that map to peptides from rare proteins. So we call this our dynamic range problem. But the reality is, this is a biotechnology problem. It's not limited to proteomics. And here we can ask, what is the smallest number of transcripts from our assembled transcriptome that we need to account for half of all of the mapped reads in an experiment? Now we can see that in some of the FASTQ files for Gupta, that we need something up to 4,000 different transcripts to account for half of the transcripts that map, or half of the reads that map, and that's pretty good. But look at these 2 samples. These were the day 3 shoots and the day 3 cotyledons that they produced sequencing experiments for. And you only need even less than 100 transcripts to account for fully half of all of the mapped reads. So that's pretty shocking. We see then that there's a huge disparity. Some of the transcript sequences that we assemble have a lot of evidence behind them, but the great majority of them do not have huge numbers of sequencing reads mapping to them. Now I mentioned before that we want to deal with contamination and completeness. So contamination means that although we thought we were sequencing reads, sequencing templates from Chia, it may be that other species had infected our Chia, for example. Maybe we have some bacteria or some fungi growing in our Chia. In a case like that, we would like to be able to use something like MC-SC decontamination to pull those out and give us the set of sequences most likely to represent Chia and not contaminating species. We also want to check completeness, and we said that the sets of genes that are expected to appear exactly once in this case in eudicots, a higher level taxonomic classification, we want to see, of these genes expected to appear once, how many of them were observed exactly once, here shown by the dark red bars, how many of them were assembled to two different transcripts, those are shown here in kind of this salmon color, how many of the genes were present, but only present in a fragmentary way, shown in this little pink set right here, and how many genes that really should be present were totally absent from these transcriptomes. Those are shown in gray. Now, before I said we were worried about Sridhar because it produced a smaller number of transcripts overall. But here we see that Sridhar is actually one of the most complete transcriptomes. It's very much on par with these other three experiments that we're including. So just because the transcriptome is compact does not mean that it is incomplete. Busco is an excellent way to check for that. So the other thing that we might be able to do to attribute function to these is to be able to compare our sequences either at the nucleotide level or after 6-frame translation to the proteins of other species nearby. In this case, we were looking at the lamiales. We had the set of Genlisia aria, hygrometricum, and so on, all these different species names that appear here. And remember, the slides for this are available in PDF form from the description. We wanted to ask how many of the protein sequences that we had overlapped well or poorly with these other species. Were they orthologous, in other words? In this case, we're looking at transcripts to ask about, if I see a transcript from Chia, is it orthologous to a transcript in these other species? This is called an upset plot. A lot of people have shown Venn diagrams with 4, 5, and 6, or 7 or 8 different circles all trying to overlap, but these things are impossible to read. So I would like to draw your attention to upset plots as a way to show these intersections much more effectively. Here we see that the most common outcome when we tried to compare the transcripts of Chia to all these other species was that we detected homology with S splendens only. So this largest set of transcripts, 3,000 transcripts here, are only orthologous between Salvia hispanica and Salvia splendens. Now, Salvia splendens is not a chia seed. This is in fact an ornamental plant, but it's in the same genus, and here we see that the transcriptome is recognizing that relationship very well. We could also do this on the protein level though, and when we look at protein level comparisons rather than transcript level comparisons, we are often able to detect homology at much greater evolutionary distances. So this, the fact that we're looking at transcripts is driving why we detect similarity to S splendens over everything else. We would also like to match motifs, domains, and protein families to the transcripts or to the polypeptides sequences that we've translated from them. So in this case, we've used protein ortho as shown in the previous slide to compare proteins, and we had interproscan to be able to look for these sequence motifs in the transcripts that we generated, typically after translating them to in 6 frames. So we're able to combine this information through some software called Zorbit Analyzer that we created as part of this study. Now this was kind of our first effort in this space. I would say that one of the better ways to try to incorporate this information into a sensible annotation is actually the transdecoder software that's designed for finding the most likely translation of each transcript sequence. I want to show you what happened when we tried to examine all these translations from Wimberley and from Sridhar and from Pleasilea and also from Gupta. You'll see that there's a fairly big area of blue here. That blue area represents the set of protein sequences that match to interpro only. There was no hit to an ortholog via protein ortho. This area of green represents the set of protein sequences that are hits to motifs in interpro and have orthologs in another protein database. So we see that that is also a pretty substantial player. We also have this tiny little splodge of yellow over at the right. What this reflects is the set of protein sequences that do match to an ortholog but do not match to interproscan. So what we see is that there's a huge number of proteins that we can see, that we can get some annotation information from interpro by, but a relatively small number of proteins for which we can only get orthologs. And of course there were a large number of these translations that hit absolutely nothing. The reason for this is that a 6-frame translation, by its very nature, is going to produce lots of translations that are in the wrong reading frame, even though we limited to polypeptides that were no shorter than 100 amino acids. So what did we get about plant biology from that? What we have here are 6 different signatures that are found in interpro that are all associated with what's called a fatty acid desaturase, Fatty acids may be produced in fully saturated forms, meaning they're all single bonds, and then later be modified by a fatty acid desaturase in order to produce something like a PUFA. So in this case, because that area was of such interest to us, we looked among this set. Now, there's a group led by Yufei Xue that had already cloned out fatty acid desaturase genes, 2 of them, FAD2, from Chia in the past, and they published this work. So we thought, let's try to find more genes, more transcripts in our assembled sequences that also correspond to the signatures of FAD2. We found that IPR 005804 was a very useful one. It matched to 37 transcripts in Gupta, 30 in Peleas-Celea, 29 in Sridhar, and 50 in Wimberley. So we thought to ourselves, these sequences are going to be interesting. In the paper itself, in the supporting information, we have 10 different enzymes of this class that have all been found across all 4 of these transcriptomes and that match 5804. So here I'm showing you one of the most compact examples, sphingolipid delta 4 desaturase, DES1-like. It's a great description because it tells us the function of this particular enzyme. That's derivable from the fact that this signature matches 2 proteins with that description. But of course, matching orthologs gives us that ability. And we can certainly blast these sequences in order to find hits for them in Tremble, if not in SwissProt, because SwissProt doesn't have a lot of plant proteomes yet. Okay, so this is great. Given that we know a signature to look for from somebody's cloned gene, we are able to find more examples like that in the transcriptomes after translation. Next, we want to be able to ask this fundamental question: if you have a proteome in this species, how well can you identify the tandem mass spectra to peptide sequences? What I'm showing here is called a heat map. So each of these 5 columns represents a different database that we attempted to identify our spectra with. MDPI is the published transcriptome that appeared in the Wimberley papers supporting information. So when experts do a transcriptome assembly, this is what results from it. This is my first time publishing any work in RNA-seq assemblies, so I was worried that I might be making beginner mistakes, so having their completed transcriptome to compare against was really good. Now, MDPI can be compared to Wimberley because this is the same set of FASTQ files being processed differently to produce these transcriptomes. And we see that if we look at the shades here, which represent the number of distinct peptides identified in each raw file, we see that the pattern is quite similar. It's hard to tell whether MDPI or Wimberley is darker or lighter. And the reason for this is that it's the same data. Our transcriptome then is probably about as good as theirs for doing this identification. But if you look at this plot, especially if you've downloaded the PDF, you'll see that this middle band for Peleas-Celea is a little fainter than the others, which is to say that when we searched our data against Peleas-Celea, we had a slightly lower number of identifications than we did from the other sets. Now, how much worse did it get? How much worse did our numbers of identified peptides get by dropping back to Peleas-Celea versus something like the MDPI transcriptome? And the answer is, we may have a swing as little as 18% difference or as big as 29% difference in how many distinct peptides we can identify. So yes, it does matter which transcriptome you use, and yet, even a relatively a relatively weak, relatively incomplete transcriptome may still be good enough to power some amount of peptide identification. We ran into some special challenges, as I mentioned, trying to work with the Aguiar-Toala dataset. These are not tryptic peptides, and they're not coming from a very complex tissue, they're coming from seeds, which means that you have a whole lot of signal concentrated in just a few proteins. So when we did this identification, it was quite clear that Pillay, Salea, and Gupta, these two circles on the side, had the largest numbers of identifications overall when we used MSGF+. So we were very happy with the results here. But we noticed that even though these were the best proteomes to be working with to do these identifications, there were a lot of peptides that were only able to be matched when we dealt with Sridhar. And we were able to find a few protein sequences that appeared in Sridhar but not in these others that were simply being missed when we tried to identify those peptides with those other two databases. So even though you might have the best of the input transcriptomes to start with, it may yet be that because it lacks particular proteins of interest, it fails to identify some peptides you'd really rather get. Okay, so that's our story on the proteomics side, that we were certainly able to support proteomics identification, and that there were some reasonably large differences. 29% gain of peptides identified is a pretty big jump by going from the worst to the best transcriptome for a given data set. You may simply have to assemble them all and figure out which one is going to give you the best identifications for your data set. We also spent a little time looking at proteins that were specific to particular assemblies. In this case, we're looking at each of the samples, essentially, that were run by Brett Cooper. We have the 1D, just LCMS-MS shot of the cotyledons, hypocotyls, roots, seeds, and then we have 3 2D fractionation experiments, cotyledons, hypocotyls, and roots. Now this dark band that you're seeing represents the set of proteins that had more than 100 spectra matching to them for particular proteins. So each of these rows represents a different protein. What we see then is that we have more than 100 spectra seen for these proteins in the seed shot, but far less in the other tissues that were analyzed. So we can easily then make the claim that these proteins all associate very, very strongly with seeds, and that helps us to enumerate the proteins that we really want to pay attention to in trying to understand this new transcriptome that we've been able to assemble. And when we do blasts of these, we see that a fair number of them match very, very well to proteins known to be in seeds from other species. So there are quite a few takeaway messages here. The first is that transcriptomes can vary considerably in completeness for a given species and even within a particular experiment. Don't forget that the data from Pillay Salea were great, and yet the same team's analysis of Veracruz or Jalisco cultivars produced far, far fewer transcripts, far less completeness in the transcriptome we were able to assemble. LCMS-MS is quite likely to sample a smaller proportion of a genome than will RNA Seq, even when we use fractionation. It's still the case that sequencing technologies that can benefit from PCR, like high-throughput sequencing, massively parallel sequencing, that they are able to detect sequences that are present at a far lower concentration than we can in proteins. We don't have a Xerox machine for producing huge numbers of copies of proteins at low concentration, and that can really hinder our ability to identify them well. RNA-seq provides a sufficient set of transcript sequences to support proteomic identification. Yes, I think that even some of the earlier transcriptomes that we saw, such as Sridhar, which was collected back in 2017, even data sets of that age at this point are still very, very good for producing databases that you can use in protein identification. And finally, maybe from time to time I should put down my burger and fries and have a nice salad that features some chia, because it's a great way to get polyunsaturated fatty acids. And here are some beautiful chia seeds for you to ponder that with. So I hope that this has been a useful journal club for you. I hope you got some ideas of things you'd like to try, and I'd be happy to hear from you if you have any more questions about this work. By all means, please pop over to MDPI Plants and have a look at their special issue on harnessing the power of genome and transcriptome assemblies. Thank you very much.